Today we are looking at biological molecules protein. Just like carbohydrates, there is a repeating monomer unit. This is called an amino acid. Note here the amino group on the left and the carboxylic acid group on the right. When these two amino acids bind together, forming a dipeptide, it creates a condensation reaction. Again, water is released and a dipeptide is formed. Note the bond here showing the peptide link or peptide bridge. When these amino acids bind together, they form something called a polypeptide, a single strand of repeating units like the necklace or chain. This is called the primary structure. Once this primary structure folds, it can form an alpha helix or a beta pleated sheet. Both of these are secondary structures. The next level of protein structure is called the tertiary protein structure. This can be either fibrous or globular protein. The one on the left is usually a structural protein. The one on the right, the globular, is usually enzyme or hormone. Looking at the fibrous proteins first, there are two we need to know about: collagen and keratin. Collagen, in the top diagram, shows a triple helix. It is a fibrous protein and is strong. The lower diagram on the right is keratin. This is a two helix structure. Note the disulfide bonds. This is extremely strong and a stable molecule. The fourth level of protein is called the quaternary structure. This is where two or more polypeptide chains bind together. Here we see hemoglobin, the most famous example. This diagram summarizes the protein structure from primary through secondary to tertiary and of course finally to quaternary. Of course, not all proteins can be quaternary, as they may be a single polypeptide chain. How are these structures held together? As you can see from the diagram, there are really four major types of bond: the hydrogen bond, the hydrophobic interaction, the disulfide bond, and the ionic bonding. Hydrogen bonds tend to occur in the secondary structure. Hydrophobic interactions and disulfide bonds appear in the third, along with ionic bonding. These give immense stability to the molecule. In particular, the disulfide bond, shown as between two cysteine molecules in green, is a very strong bond, particularly at the tertiary and quaternary level. Here we see the interactions in a single polypeptide chain as it is convoluted over into the secondary and then tertiary structure.